everyone to migrate from the waiting room to our main session here. If I haven't met you, my name is Ken Shopman. I'm ABRF's Executive Director. Thank you for joining us for today's program. And thanks to our friends at ClaretBio for hosting. I'm just going to go over a few housekeeping and other announcements and updates from ABRF. Feel free to introduce yourselves in the chat. ABRF is a community. We like to see who's joining us from around the country and even around the world. Your name, your institution, where you're connecting from. Please also use the chat if you have questions. Our presenters will stop for questions during the program and afterwards, so that'll be a good way for those questions to get organized. And if you have suggestions for other topics you'd like to see ABRF present in these online programs, please send us a note, abrf@abrf.org, and we'll be happy to pass that on to our Education Committee and Membership Committee for future programs. Just some updates and announcements. Tomorrow, we have the next in our very exciting series of programs on using AI tools in core facilities, specifically in imaging facilities. So tomorrow's program is going to focus on some of the commercial AI tools that are available with demonstrations and content from some of those commercial providers. You can see the QR code on the screen if you'd like to register for those programs, which will continue monthly for the rest of the year. And then later next week, we have our monthly virtual peer mentoring conversations. So if you have a question or a challenge that you're facing in taking the next step in your career or with a specific part of your work in core facilities, your ABRF colleagues are some of the best resources available. So these conversations are really informal, comfortable ways for you to ask questions and get feedback and input from your peers and colleagues. And the next session is next week. Once again, you can follow the QR code to register for those. I hope you're participating or planning to participate in some of the upcoming chapter events. October is a very busy month for our chapters, and we've even extended into November with our special West Coast chapter meeting in November in La Jolla. So you'll find all of these chapter event information and registration links on the ABRF website under chapters, regional chapters. So if there's one of those that's close to you, please take a moment, sign up. These are very accessible, reasonably priced events, and it's a way for you to connect with other colleagues that are in your neighborhood, so to speak, or in the greater region, and also with those technology providers that are nearby. Often the regional offices will send their local representatives, so chances are it's the person that's helping you with that instrument at your facility, and you can get some concentrated time with them as well. I hope you're participating in the core community, which is an online environment where you can connect with all 3,000 ABRF members in general conversation or in very targeted conversations around topics like biobanks or electron microscopy or protein production. So no matter if it's a broad topic or a narrow topic, you'll find colleagues in ABRF in the community that are working in that area. So it's a great way to post questions, share resources, ask for help, and connect with one another. If you have any questions about getting involved in the community, once again, please reach out. We're happy to walk you through that process. If you participated in these programs before, you know that we do this very quick one-question survey. We want to know what other organizations you may be a part of. ABRF is in active conversations with other peer societies to form collaborations that will result in future programming, both at in-person events and online. But the more we know about the other organizations and interests that you have, the better we can tailor and target those collaboration efforts. So take a minute, it's very quick, follow the QR code or go on, use the link to respond. Here's our agenda for today. And with this, I will turn it over to our moderator, Dr. Varsha Rao with ClaretBio. Varsha?
Thank you, Ken. And thank you everyone for joining. Please bear with me while I share my screen. As I said, thank you everyone for joining. We are happy to co-host this webinar titled Tackling Challenging Samples for NGS Application. First of all, I'd like to thank ABRF for giving us this opportunity to co-host this session. I'm hoping that you'll take away very useful insights about what ClaretBio's products can do in handling highly degraded samples, challenging input types, which are very often encountered in core facilities. Very briefly about what we do at ClaretBio. We are a startup that is based in Santa Cruz, and over the past about 7 years, we've developed in-house expertise in developing challenging tools for NGS application. Tools for challenging input types in NGS applications. Our core product is called Seriously. It was launched in 2019, specifically focusing on liquid biopsy samples, so that is cell-free DNA. We do focus on fragmentomics, and we can generate libraries from input types such as plasma, saliva, and urine. And I know Dr. Neeti Swarup is going to cover some of these applications in her talk. In 2022, we expanded the application of our core product by developing a fragmentation module. So this allows you to generate libraries from genomic DNA, even from FFPE with DIN values less than 3. This generates highly complex libraries, works very well with downstream targeted enrichment applications. And then finally, earlier this year, we applied our single-stranded ligation approach to RNA-seq. What this does is allows you to make directional libraries without second-strand synthesis and without needing any repair module upstream from FFPE RNA samples that are highly degraded. And this product is called Really RNAN. It does ribodepletion. With that, I would like to move on to the session today. If you have any questions in general, do write to us at info@claretbio.com. I'd like to now introduce our speakers, Dr. Jun and Dr. Neeti Swarup. The flow of today's talk is going to be Dr. Jun is going to speak first. Dr. Jun is currently the director of the genomics core at the Texas A&M University. She received her PhD from UC Davis in 2009. Her expertise spans from the early days of Sanger sequencing to NGS technologies and the current state of art 10x spatial and in situ technologies. She is a member of the ABRF and she's a part of the genomic sequencing group. And today she'll talk about her work done at Marshall University. Dr. Neeti Swarup is a research scholar at Professor David Wong's lab at UCLA. She received her PhD from Seoul National University from South Korea, working on oral cancer. Her expertise spans liquid biopsy, oral pathology, to name a few. At UCLA, she leads multiple projects as the co-investigator on awards granted by the American Cancer Society and the National Cancer Institute on novel liquid biopsy markers in oral cancer and in the development of assays for ultrasensitive ctDNA and microRNA detection. And today she'll talk about her work on ultra-short ctDNA. We will first have Dr. Jun Fan speak to us, followed by Dr. Neeti's talk, and then we'll take questions at the end. With that, I'd like to invite Dr. Jun to begin her talk.
Thank you. Go to my sharing my desk. Can you hear me? My slides?
Yes, we can see it.
Okay. Thank you for introduction and thank you for inviting me to give today's talk. The talk I was discussing today was completed at the Marshall University last year. So currently, I'm working at the Texas A&M Institute for Genome Science and Society. Before I give the talk, I would like to give a quick disclaimer. This talk is only for the research communication purpose. I have no financial interest in any commercial product mentioned and used in this presentation. Today, my talk is about the DNA library preparation from the ultra-low, high degradation genomic DNA sample. To give a quick overview, I will start with some background on this study, how it got started. And I will walk you from the library preparation protocol, followed by the library enrichment and sequencing. Then I will wrap up with a little bit of, briefly, like the downstream data analysis. This study began as a collaboration among 3 groups: Marshall Genomic Corps, Marshall Forensic Science Center, and a company called Parabon. The Parabon is a DNA technology firm. It is cutting-edge snapshot advanced DNA analysis platform. Is a game changer in DNA forensics. It's widely used genetic genealogy, phenotyping, and kinship inference. The goal of this collaboration is to introduce next-generation sequencing technology into forensic field. The study material we worked with was artificially aged bone sample. The reason we choose this type of sample is to match what you will find in real forensic case. To generate like aged bone sample, first we took fresh bone sample buried in soil inside the glass container. Then this container was placed into the incubator, which was set to 65 degrees. After 2 to 3 weeks of incubation, the bone was pulled, cleaned, and then stored in the freezer until it's ready for the genomic DNA extraction. The genomic DNA extraction was fully automatically on Agilent, the QIAGEN EZ1 advanced instrument used the QIAGEN kit EZ1 DNA Investigate. This instrument run a pre-programmed protocol started on a car. Once the car inserts in instrument, the process runs smoothly according to pre-programmed setup. Up to this point, the Marshall Forensics Center handled everything: the bone treatment and the genomic DNA extraction. The rest of the lab work were conducted by Marshall Genomics Corps. When Marshall Genomics Corps received the DNA sample, the first thing we did was check its quality. We checked it on Agilent tab station using Agilent genomic DNA screen tab. We got the DIN value of 2. The DIN stands for DNA integrated number. It's a metric used to access the DNA degradation with a scale 1 to 10. 10 being perfectly intact, and the 1 being badly degradation. A number of 2 means like highly degradation DNA sample. In this imaging, we can see the DNA size distributed from 100 base pair to the 4,000 base pair. Before we move on to the next step for DNA library preparation, we trim down the molecule, these samples. We did on covariance ME220. And this condition, we check like the fragment, the molecular by Agilent bioanalyzer with a high sensitivity kit. In this gel-like the imaging, we can see most like a molecular lower than 500 base pair, with majority around 300 base pair. Maybe this one can show give a better visualization. Here is a standard DNA library preparation protocol. There are key steps, several key steps involved: end repair and A-tailing, followed by adapt ligation. A-tailing increase efficiency of ligation, adapt ligation. The P5, P7 sequencing adapter are introduced by PCR amplification. This is the final library structure with the P5 at the 5' end, P7 at the 3' end, library insert in the middle. This protocol is currently is commonly used. However, this method only can convert double-strand DNA into sequencing-compatible molecule. However, our DNA samples is a mixture of DNA with a single-strand DNA, double-strand DNA, also damaged double-strand DNA. So this method doesn't work for our purpose. We need a method can convert all, like single-strand DNA, double-strand DNA, as well as damaged double-strand DNA, all convert into sequencing-compatible molecule. That's when we thought about single-strand DNA library preparation kit. That's all the CS kit pop up. Here is a workflow of a series kit. In the kit, there are thermostable single-strand binding protein involved. This binding protein keep like the template DNA in the single-strand status. Also, the forward and the reverse adapter, they are double-strand DNA. Sorry. Okay. This double-strand DNA, in the bottom strand, they have random primer overhangs. This spring adapter help maintain the correct direction of the adapter. Unlike the standard protocol we just mentioned, this adapter ligation directly to the molecule without the ending polishing needed. So this one, they can preserve the native end of the molecule. Also like the P5, P7 sequence adapter introduced by PCR. We figure out this kit would just what we needed. We also thought this kit might give us better molecular recovery and better library yield. So we decided to use this kit to make a library. We choose the pickle kit with the 3 different amount input: 1 nanogram, 500 picogram, and 50 picogram, 50. Since our molecule was already fragmented, we begin with the adapter ligation step. Then bead purification, we choose the larger fragment module. Next, we end the unique molecule identified by primer extension. In the final PCR amplification stage, based on different amount input, we adjust the PCR cycle with 11 cycle for the 1 nanogram sample, 13 cycle for 500 picogram, 16 cycle for the 50 picogram. The bead purification, we choose the single-strand bead purification, also use the larger fragment module. Here is a final, our library structure. The P5 at the 5 end, P7 at the 3 end, and the molecular in the middle. This structure just look like the one made by double-strand DNA library preparation method. So it's fully compatible with the Illumina sequencing platform. We checked this library quantity and quantity. We measured the library concentration on the qubit with a qubit double-strand DNA high-sensitivity kit. The DNA yield range from 170 nanogram to 330 nanogram. The library check on the bioanalyzer. You can see there are strong broadband between 300 base pair to the 500 base pair, all across the three samples. Since the study material was buried in the soil and might have a bacteria and the fungal contamination, we decided to enrich this library using a human exome panel. We choose the ABRF Bioscience MyBait kit to do our enrichment. In this kit, they use the biotiolated probe to capture the NGS library. Then the captured library pulled down by streptavidin tagged beads. After several steps, the washing removed the uncaptured library. The final library went through PCR. This our enrichment method. We choose like the standard module with the hybridization temperature at the 65-degree, incubation about the 20 hours. The final PCR cycle, we choose 12. Again, we check this library on bioanalyzer. You can see strong band between 300 and 300 base pair to the 500 base pair, all across the three samples, ignore like this. So this library looks pretty good on bioanalyzer trace, but the complexity of the library can only be evaluated after sequencing. So we promise the sequencing this library over on our NextSeq 2000 using P3 flow cell, configure it for 2x150 pair and arrays. The sequencing run went pretty well. You can see here, the AGBT nucleus are balanced evenly across all 300 cycles. This one is just index spiking. And the average Q30 score for this run is above 90%. We received 1.3 million past field arrays. Up to this point, the lab work was complete. Next, we send the FastQ data to the Paragon, and the Paragon team conducts the downstream data analysis. Here is a sequencing result provided by Dr. Gritek from Paragon. Each library we received over 755 million reads. This read was aligned with the human genome, achieved more than 95% alignment for each one. Also, this read was used for genome call. We were able to identify more than 88.7% of the SNP call out of the 2.2 million total. To further access the quality of our NGS data, our data was compared with previous data set that had already been verified by Paragon. This previous data set was generated from the same DNA sample, but use a different method and with a different amount insert. S5, S8 microarray data with a 10 nanogram input. LIFM0014 is another NGS data, use a different NGS library preparation method with a 1 nanogram input. All these sleep call from all this data were combined into one file. Then the Parabon team conducted a pairwise identity by state comparison. Here is the result. You can see our NGS data are highly consistent with the other data set. Look like the microarray data or another NGS data set. So based on like that, we made a conclusion like the ClaretBio CSLA library preparation method can effectively recover degradation of DNA and support high quality NGS analysis. So at the beginning, I mentioned our, this study is focused on the forensic field. So Parabon team applied all the data into it is a Snapper advanced DNA analysis platform to make ancestry and the phenotype predictions. Here is the ancestry prediction result. You can see they are highly consistent and narrow down northern eastern ancestry. The pigmentation phenotype prediction were classified into 5 groups. Also, they highly coordinate as well, right? Even though there are some minor variance in red hair and freckling, the overall conclusion are same. Again, the face predictions also line very well among this data. Still, there are minor difference there, but nothing changes the overall interpretation. I think the slides pretty much speak for itself, so I would not dive into the detail. Also, this result comes from the Parabon. I'm not expert here. Take-home message: the CSLA kit can deliver accurate outcomes with as little as 50 picogram of input DNA. Acknowledge. I want to thank Dr. Primarino, Dr. Salisbury for their support. Without it, I cannot complete this study. I also want to thank Lauren Tyler for providing DNA samples. Special thanks for the Parabon team for their downstream data analysis, especially Dr. Ellen Greatag for her help. Tyler currently works at the Wisconsin Department of Justice. Thank you. I may take a question later.
Thank you, Dr. Fan. I just wanted to make a quick announcement about questions for Dr. Fan. So we'll take questions at the end of the session, but feel free to post your questions in the chat. I see that there are people who've posted questions already. If there are technical questions about Seriously, we'll try to answer them today on the call. Otherwise, do write to us with technical questions. And then finally, one housekeeping announcement that I missed at the beginning was we will have a survey at the end of the talk. This gives you an opportunity to also get a 50% off kit, a Seriously 24 reaction kit, and also we'll be doing a lucky draw to give a free kit away along with some ClaretBio merchandise. With those housekeeping announcements, I'd like to invite Dr. Neeti Swaroop to give her talk on applications of cell-free DNA.
Hello, good morning, everyone. I hope everyone can see my screen and can hear me properly. So as Dr. Rao already introduced about me, I am a research scholar at UCLA School of Dentistry. I work with Dr. I work at Dr. Wong's lab, and our main focus is liquid biopsy, and our biospecimen of interest are basically plasma and saliva. So I'll be talking about expanding horizons of cell-free DNA-based liquid biopsy. So at the present is a very exciting time for liquid biopsy, especially with the discovery of cell-free DNA and identification of fragmentomic features, which Dr. Rao introduced during her seriously introduction. And along with the other analytes that people have previously seen, like circulating tumor cells, RNAs, and circulating tumor DNA. So our lab is mostly concerned with the bioanalyte of cell-free DNA and circulating tumor DNA. So as we know, there are a lot of analytes within the liquid biopsy domain that people are interested and have been very focusing on. Whether it exosomal assessments, or whether it's circulating tumor cells, whether it's microRNA, whether it's proteomics, metabolomics, all these have given a lot of insights. But the most interesting one that has emerged in the most recent times is cell-free DNA. Interestingly, cell-free DNA has been discovered quite a long time back, but its clinical relevance has got a lot of center stage in the recent times, be it prenatal diagnosis, be it cancer detection, be it autoimmune detection. So our lab focuses on sequencing, genomic sequencing, and non-sequencing based assessment of cell-free DNA. So what our lab here has achieved with is the identification of different subsets of population of cell-free DNA. So we utilize a particular, an interesting workflow wherein we try to harness the shorter pieces. So as we have talked about the degraded DNA samples, DNA molecules to be extracted from the samples, we find that from our workflow, we identified that there are very short pieces of cell-free DNA that circulates within plasma or saliva or urine. And these are harnessed basically from two main steps. So how we have modified the DNA extraction and how we use the single-stranded library preparation to go ahead with the whole sequencing pipeline. So as you see here, the ultra-short cell-free DNA that we call is around a 40 to 70 base pair length cell-free DNA fragment. And in saliva, we see that the DNA is very highly degraded, and we see a regular 10 base pair breaks with the peaks and the troughs as we see here. So this makes cell-free DNA a very challenging sample, a very challenging analyte to be harnessed, whether it's from plasma or saliva. So we call this pipeline as broad range cell-free DNA sequencing because we get the more routinely known mononucleosomal or monosomal fragment of cell-free DNA, which is around 167 base pair. And along with that, we harness this newer subset of population that is ultra-short cell-free DNA. So before we proceed further, I would also want to include this topic here is that apart from circulating tumor DNA detection, so people have recently involved topological analysis or epigenomic analysis in their cell-free DNA. And with that in place, what we have tried to harness is with the low coverage sequencing and harnessing these shorter pieces of cell-free DNA, we try to do a more topological or a morphological assessment of cell-free DNA. So apart from conventionally known mutation detection, SNP detection, CNV detection, we do a fragment length analysis, topological analysis, and more recently we are delving into epigenomic analysis as well. So the sample here I will be discussing is actually saliva. And as we know, we can harness variety of biofluids from a human, be it plasma, blood, cerebrospinal fluids, saliva, urine, tears. People are trying to do with pleural effusions, gastric lavages. So with all those in place, saliva becomes most easily accessible However, saliva comes with an inherent challenge of being the biofluid which is exposed to the environment continuously. And along with that, oral cavity, saliva being present in the oral cavity, it is very much enriched within the microbiome. So it brings with it a significant set of challenges. However, we try to address them from our analysis and workflow standpoint. So here I would like to discuss on how we have harnessed saliva and how we use saliva to employ the broad-range cell-free DNA sequencing to see how we are able to harness the cell-free DNA molecules from saliva for our cancer detection disease model. So first, we need to see that saliva in itself is a very complex biofluid. Because as we collect the saliva, that's one of the biggest challenges we begin with. Because saliva is enriched with a lot of immune cells like lymphocytes, neutrophils. They come within the saliva. There are also shedded keratinocytes within the saliva. So with this in mind, we try to classify the saliva as like a mixed type of analyte in which the cells are included within the supernatant. And then as we go routinely know 23andMe, so it's like a swab of oral keratinocyte or the cellular pellet that we get from a saliva point, from the saliva, and use that for the genomic DNA analysis. And the most cell-free type, which is the supernatant, and is most exclusively for like fragmented or degraded DNA fragments in the supernatant component of the saliva. So we tried a bunch of saliva collection methods, ranging from just spitting in the tube, and then we have a standard operating procedure of how we spit and centrifuge the tube to collect the supernatant. There are saliva collectors in the market like Oasis, Spectrum, Zymo, and Ora Collect, and try to see and compare. So our main focus here is for cell-free DNA from saliva for broad-range cell-free DNA sequencing. So we basically go for the SOP collection method, which is more or less towards a cell-free DNA, and the Spectrum, which has a lot of admixed genomic DNA in it because of a lot of cellular components within the saliva sample. So extraction of salivary cell-free DNA. So at the present, we are following a silica column-based extraction from our pilot experiments where we try using magnetic beads and silica columns. So here we have employed the QIAGEN circulating nucleic acid kit. And so this is our publication where we first describe on how ultra-short cell-free DNA can be observed. And it uses a silica-based or a magnetic column-based or magnetic bead-based. And the silica column, the reason we have a modified protocol for the ultra-short recovery is the application of more DNA-phobic buffer. We are working on the details of how this is made possible, that when we are adding a little more of DNA-phobic buffer, how it basically enhances the recovery of the shorter fragments. So we call them the QIAGEN Classic and QIAGEN microRNA protocols. So here, this is the tape station Agilent 4200 tape station result we use with the cell-free DNA and tape. And we were able to see that our genomic content within the Spectrum saliva-based collection was extremely high. So with the Spectrum, we see somewhere over 10,000 of the genomic DNA. So our whole DNA extraction is more biased towards the genomic and the longer fragments of the DNA. So our focus is on the shorter fragments of the DNA. So we see here with the standard operating procedure, although our yield is lower, the overall assessment that we see that the cell-free DNA that we get is better. It's slightly better with the standard operating procedure. So with comparing both the QIAGEN microRNA and QIAGEN classic protocol, so this is actually post-sequencing results that we are showing here. However, we see that even when we make the libraries post-DNA extraction, we find that the QIAGEN classic protocol basically enhances the longer recovery more as compared to the shorter recovery. So we keep our workflow more inclined towards the QIAGEN microRNA protocol. Then we come on to the library preparation and how we prepare this DNA going forward. So we first compared like if we should just use a standard double-stranded library preparation or if we should use a single-stranded library preparation. We have used the Claret for the single-stranded library preparation, seriously nanoplus. And for the conventional double-stranded library, we have used the NEB Next kit. So with the use of both the library preparation, we were actually able to see that there is a possibility of very mixed population of cell-free DNA within saliva. So I would show some results here that we got from single-stranded and double-stranded library preparation. That in the single-stranded library preparation as compared to the double-stranded, we have a more uniform presence of the cell-free DNA throughout all the fragments. So here we have not done any analysis, just the recovery of the cell-free DNA fragments based off of length. So we see that it's a more uniform recovery throughout. However, when we tried doing some experiments with repairing the DNA, especially the nick repair, we find that the nick repair just enhances the longer pieces a little bit. However, there is a dramatic impact on how the double-stranded library behaves when it is nick repaired. So suggesting that the shorter pieces are basically the double-stranded DNA is actually leaving out a lot of nicked double-stranded DNA, which are longer in length. So this made us to go on with the workflow where we use the single-stranded library preparation for our cell-free DNA. So for our broad-range cell-free DNA sequencing, we use this protocol wherein we have the DNA extraction with our column-based extraction. And we use a seriously single-stranded UMI-based library preparation. Then we go on for the our bioinformatic analysis pipeline. And as I mentioned previously, we don't go for mutation-based detection. We are looking more for morphological or topological features of cell-free DNA. So being a challenging specimen that saliva is, especially due to the microbial presence within the saliva, the pipeline that we follow is slightly different from the pipeline that we have for plasma. So for our quality control in bioinformatics, so this is our in-house developed pipeline that we use. And post alignment, we filter out all the unmapped reads. So when we align it to the human genome, we find our primary alignment with the high MapQ qualities is only around 70%, and the remaining ones are left unmapped. So for that, we go for a secondary alignment where we try to align it to the microbial databases that are out there. And we again try to align with the human along with the microbial database. And that's how we basically do the alignment, our processing for the cell-free DNA from saliva. So here we have used our salivary cell-free DNA for the gastric cancer detection disease model. And I would like to show some results here. So as you can see for the salivary cell-free DNA, in general, we can see a dramatic difference between how the fragments are seen between the cancer and the non-cancer disease, non-cancer cohorts already. However, the topological, morphological, and other features that we see include the jaggedness profile, how the microbial population is there, how the DNA is cleaved at different sites, how much is the mitochondrial contribution, how is the genomic spread, how is the fragmentomic profile of the salivary cell-free DNA, or how it spreads across the human autosomal chromosomes. So for the jaggedness index, as we have seen previously compared between plasma and saliva, saliva has a more jagged profile with very pronounced peaks and troughs. And these peaks and troughs are very regularly placed between around 10 base pairs. So they have been correlated, this 10 base pair peaks and troughs, with the linker portions after the mononucleosomal DNA. So with the linker portions and the degradation away and aside from the linker portions, we see the pronunciations of the peaks and the troughs. With this 10 base pairs peak and trough, we tried to see if the variation between the jaggedness profile is different between the cancer and the non-cancer specimen. And we were able to see that it was very different as compared between both the cohorts. As we see that the saliva from a non-cancer cohort was more jagged as compared to the cancer cohort. Then the most routinely reported fragmentomic analysis, which even Seriously Kit is designed for, essentially, is the fragmentomic analysis. So it has been reported that the cell-free DNA is more fragmented or shorter within the cancer as a disease cohort. So we wanted to see how the cancer cohort presents in our proof of concept data. And there we try to see the fragmentomic profile of the short and the long ratio for the salivary cell-free DNA. So we see a more interesting profile than people have observed for plasma, where if we are seeing the cancer cohort, the cell-free DNA is more fragmented. However, we are seeing the reverse here, where the non-cancer cohort is more fragmented. So we have actually observed in our discussion, and we want to do some experiments, by including some microbial analysis in it. So the reason we see such kind of a reversal in the profile here is mainly we are attributing it to microbial activity on the cell-free DNA and how the microflora changes within the cell-free DNA from gastric cancer and how that affects the entire salivary cell-free DNA spectrum, affects how our salivary cell-free DNA looks like post-sequencing. So we see that it has been a very consistent feature. We actually even scaled up the study and were able to see this kind of results again. Then we talk about how they are spread across the human autosomal chromosomes. And we find that the contribution of the salivary cell-free DNA that we see from human autosomal chromosomes within the cancer and the non-cancer cohort was significantly different, and especially when it comes to the telomeric portion of how the non-cancer was more from the telomeric portions than as compared to the cancers. Then other factor that we are very much looking into for salivary cell-free DNA analysis is the mitochondrial contribution. And with the mitochondrial contribution, we have the metric that is in place is mostly about the bulk of the mitochondrial cell-free DNA that we see. And we see that the mitochondrial bulk within the cancers increase as compared to the non-cancer cohort. Then is the genetic identity. So by genetic identity, we mean which gene or genomic element our cell-free DNA reads aligned to. So we find that the peaks that are formed, like the tendency of the reads to align to a particular portion, is more in terms of non-cancer when compared to cancer. And when we try looking at different elements, be it exons or introns or promoters, we see that there is a significant difference of how they align to different portions. So this is something where we are actually looking on whether it is actually a contribution from different genes that are present as a gastric cancer carcinogenesis genes, or is it only a representation due to the nuclear fragmentation of the DNA cleavage within the saliva. So here are listed out some of the significantly different genes that we have identified in different elements, be it promoters, introns, or the intergenic portions. Then comes the most interesting part, is the microbial contribution to salivary cell-free DNA. So at present, we have harnessed the one codex database, and we have used the Metaflan database to analyze different microbial species and classes which have contributed to the microbial cell-free DNA bulk within the salivary cell-free DNA. So it has been noted that the Haemophilus species and Staphylococcus species, they release certain nuclease enzyme which help them in forming the biofilm. And we have also observed that the tendency of certain Haemophilus species was different in contributing to the bulk of the microbial cell-free DNA. So the reason this is very important, and this needs further looking into if we change the microflora, how the cell-free DNA behaves, is because the dynamic nature of the oral cavity and how the oral microflora shifts in different disease types and how oral microflora shifts from person to person as well. So this way, it is a more challenging thing of how we take care of the microbial cell-free DNA read. This is one of the reasons that we have the primary, when we try and align for the human genome, we only go for the high quality and completely mapping reads only for the human genome. And then we discard the rest of it because the microflora and the microbial contribution to the bulk of cfDNA can actually be variable. So that is why this thing actually needs more and more looking into. And then this is another DNA feature that people are looking into is the end motif profile for cell-free DNA. So the end motif profile is basically the portions within the DNA which are susceptible to nuclear cleavage by different DNase enzymes. So we find that the nuclease, the former end motif due to the result of different nuclear cleavage are different between the two groups. And this can be a result of the nuclease activity from the human himself or herself, or the oral microflora nuclease activity. So then we end up trying to analyze all these features because the data is very big. We try to do the principal component analysis and we see that all these features in terms of obtaining the salivary cell-free DNA is very helpful in classifying gastric cancer from non-cancer. And then we also tried seeing if we can reproduce these results as well. So the data that I showed was from the pilot data set, and we are currently working on a validation data set of around 110 samples. And we see that the fragmentomic profile and the end motif are kind of reproducible. So to conclude, the salivary cell-free DNA is actually a very interesting and a promising analyte to be used in the liquid biopsy spectrum. However, how we harness the salivary cell-free DNA is something that needs very careful investigation. So with our workflow, we feel that using a single-stranded library preparation kit, such as Seriously NanoPlus kit, has actually helped us harnessing the cell-free DNA molecules more effectively than the conventionally known processes. And not just the bench process that is involved in tackling saliva as the biospecimen, it is also the bioinformatic analysis that we have to put in place for salivary cell-free DNA analysis. It's because of the more complex nature of the salivary cell-free DNA. So here are some of the resources and the publications that we have from our group. So we have also, as mentioned before, we have delved into epigenomics, and our paper was published this year in nucleic acid research. For that as well, we have utilized the Seriously NanoPlus kit, and we have they have been very helpful in providing us with adapters, and we have published our pilot results. We have used this pipeline in plasma and published with a lung cancer disease model and the resources from our lab. And these are the people who have worked together in developing all these features, the bench processes, the bioinformatic processes, and the funding sources. Thank you. I can take some questions now.
Dr. Neeti and Dr. June, we have now opened the session for question and answers. I request the audience to ask their question either in the chat or in the or live. And if you do have other questions, do write to us. We'll pass it on to the speakers, or if we can answer them, we will do so. We will also share a recording of their talks. It'll be available as a resource on the ClaretBio website. As I mentioned earlier, we're planning to do the survey. Please use the QR code that is provided here to answer that survey. As I mentioned, this will give you an opportunity to get a 50% off kit, and also will enter you automatically for a draw to get a free kit and a t-shirt. You can also meet us at these various events, MVACT, ASAG, and AMP. I can read out a couple of the questions. I think one of the questions was, how do you handle human endogenous content? How do you prevent the presence of endogenous content in your final library? Dr. Jun, could you answer this question, please?
Yeah, actually, I think that Kelly already answered the question pretty much very well. So, like, the only thing I can say, beside what she mentioned, the only thing I added is like, just when you do the practice, be careful. I never open like the reagent bottle or any of the, the stuff like that, longer than should be like that. I never open, that's like what I say, just always like when you transfer reagent or something, you close like your tube as soon as possible. So try to avoid it in your practice, in your practice. That's I can say. Besides like we do need like the positive pressure in the room and limited the people that get in the room, as just do everything you can do to prevent the contamination. And also, I see like the Kelly's question about like the percentage, like that one, actually she didn't miss anything. We didn't do it. We just like after the library preparation, we just directly go to the enrichment. So we don't have like that data.
I see Berkeley has joined in. Did you have any follow-up questions on that, or can we move on to the?
I wasn't sure if she was responding to my question just now, the one that I posted, or not. But essentially, like, we've had a couple of years of experience using this kit, attempting to get sequence information from ancient samples, and we've had a very difficult time with contamination, large amounts of modern human contaminating DNA, which we think is probably from libraries just kind of floating around in the air in the lab, from other modern samples where we make libraries. So wondering if you have any suggestions as far as separating sort of pre versus post PCR, if you have any standard practices for that, or if you have any practices for having a extremely sterile or isolated environment for the first few steps, and like what are the key steps in that process to prevent or minimize contamination from things other than your
So we have some workflow that we have other users, customers that apply, and we have used internally. Perhaps we can share some of that resource with you, Berkeley. Having separate pre and post PCR spaces, cleaning surfaces with bleach, not using the same kind of tips and so on for the steps that you do. So there are certain tips and tricks. And also, I was going to add that what Dr. Jun did in her work, where they did downstream targeted enrichment, that would help with eliminating bacterial or say plasmid contamination. And perhaps there are bioinformatic solutions to incorporate downstream after the libraries have been made to separate between ancient and modern DNA. But I can reach out to you, we can schedule a call and go into more details of how this can be achieved. I think there is another question. This probably is for Dr. Swaroop from Samantha Ortiz. For the ultra-short cell-free DNA prep, how did you help mitigate adapters or index dimer interfering with sequencing and data processing?
Most seen that if the cleanup effectively tackles the whole adapter and the dimer interference. So if we match the input and with the cleanup proportions, we basically don't get a lot of adapters. So we try to do this QC during our tape-station step, and we have observed this quite frequently. However, we can try more cleanup steps and see how it affects. And of course, we have some downstream bioinformatic steps that we try to remove all the adapters from our bioinformatic file. So we just focus on the main human aligned or the microbial aligned files.
Thank you, Dr. Swaroop. My response to this question, again, we do have a lot of in-house expertise with handling adapter dimers and sorting these kind of data. I'm happy to provide additional resources to answer those questions. I think we're reaching the end of the session. I would like to thank our speakers for graciously agreeing to be a part of this session. I hope this has been, informative to all our, attendees, as it has been for us. As I mentioned before, we'll share this content through our mailing lists, and we'll also put this on the ClaretBio website. Do take our survey, do reach out to us. We'll be able to help you with your questions and any challenges that you have with your sample types.
Well, thank you, Varsha. Thank you, June. Thank you, Niti. And thanks everyone for joining us. We hope that this was helpful. As Varsha mentioned, we will be sharing the information to everyone who registered for today's program, posting it on the ABRF site, likely on the Claret site as well. And we hope we'll see you, if not at tomorrow's program on AI and imaging facilities, at another upcoming ABRF event very soon. Thank you all very much and have a wonderful day. Bye now.